I don't do it like every day. I do it maybe once at maximum, like twice a week. It's just too much complication. But you get really muddy, and you gotta. There's a bike ride involved. The river has to be not crazy. In the summer, there's frequently a lot of thunderstorms that roll in at the time that I'd be leaving Genelia. So that's always uncertain. You don't want to be on a river in a thunderstorm. So yeah, there's a lot of things that you have to think about. Yeah, I mean, the river actually can go up 20 feet if there's a big rainstorm, because it's, just absorbing a lot of water coming, from upstream. So lots of interesting situations.
But that must be a super exercise.
Oh yeah. Oh, definitely. Oh, I mean, by the time I get to work, I'm like, but it's wonderful. Oh, wow. It just feels so good. It really does.
All righty. What are we doing? How are we doing with time? Okay, I think we're doing good with time. So welcome everyone to our monthly KNAW Dutch Biophysics seminar series.
If you click on the...
What happens? Okay, I heard something, but it's no longer there. Okay, so welcome again to our Dutch Biophysics webinar series. We have two really great speakers today. I'm very much looking forward to it. Just to tell you again the setting, we have two talks of each 15 to 20 minutes with ample time for questions afterwards. And afterwards, there is a tutorial that takes is 45 minutes with 50 minutes for questions, but that's much more interactive. Here within the visible Zoom room, we have the two speakers present, Jennifer Lippincott-Schwartz and Mike Hansen, but also three panel members. So it's Kristen Grusmeijer, who has recently started her group as a tenure tracker at TU Delft, and she's an expert on high-res super resolution imaging. We have Hilkje Geertsema, also recently started as a tenure tracker at the TU Delft in super resolution imaging, and also with a focus on education, which I guess for her, Jennifer, is a particularly good example. And then also we have Peter Zijlstra, who has established a group in Eindhoven University on like non-labeled imaging. I think this is what I know about your group. So then we of course have the other organizers. There's Chase, who's really our hero in making sure this all works. Chase Broeders from the VU, and Tom Schmitzo is there from AMOF, and I'm Lido Laan from the TU Delft. So let's start. So today we start with a talk of Jennifer Lippincott-Schwartz, and I thought about how to introduce herself. How should I introduce Jennifer? I mean, I think she needs very little introduction, but I went to her bio and I thought it was just great that if I read her bio, it starts about how she was in high school and she loved wilderness, and that actually she got, as a present for her high school diploma, apparently she got a wilderness tour of 5 days where she started to scare off grizzlies. And what I took from this is I think it just says so much about Jennifer always looking for a challenge and wanting to look for the boundaries. So I thought, I thought this is a nice way to introduce her. Now Jennifer is a senior group leader at Genelia, at the Genelia Institute, and also I think has started up some large parts of it. So I think I don't want to take more of her time. So I'm very much looking forward to the talk of Jennifer. So please go ahead. And thank you very much for coming.
Oh, yeah. Well, thank you so much for inviting me. And I'm looking forward to this interactive format. So I'm going to be talking about generally about how we as biologists can use imaging approaches to get greater insight into understanding what's going on under the hood of cells, if you will. And my talk is going to span from single molecule dynamics to whole cell organelle reconstructions. This first portion of my talk, however, I'm going to be focused primarily on whole cell EM organelle reconstructions. In my second talk later this morning, I'll be talking about the single molecule dynamics aspect. But let's start with just looking at eukaryotic cells. So eukaryotic cells evolved about 2 billion years ago. And one of the things that's really amazing about these cells is that they all are similarly subcompartmentalized. So every single eukaryotic form on Earth has associated with it the same set of 9 organelles that extend from the nuclear envelope as the ER into the secretory pathway, which includes the Golgi apparatus, transport vesicles. But there's also organelles involved in uptake, lysosomes, endosomes, and degradation, and energy metabolism. And if you look by electron microscopy, as shown in this slide here, at either a mouse fibroblast or a small little parasite that's many times smaller, you can see that the same set of subcellular organelles are found in both structures. Now, as I mentioned, these organelles are playing out very specific types of activities within cells. And this is what makes eukaryotic cells so robust. They have their own secretory system that emerges from mRNA that's being translated on the surface of this very amazing organelle called the endoplasmic reticulum. I'll talk a ton about that in my second talk. The endoplasmic reticulum synthesizes, processes, and then exports those proteins into the secretory pathway where the first organelle that picks it up is the Golgi apparatus, which further processes these molecules and then ships them off to different final destinations, which can include the plasma membrane, endosomes, lysosomes, the like. Mitochondria are super interesting organelles as far as eukaryotes are concerned, primarily because they are derived from bacteria, actually. So mitochondria are not, the thinking is that the eukaryotic cell emerged from a archaea eating a bacterial cell. And the bacteria still lives inside the host, in the case of the eukaryotic system, serving as mitochondria, an additional energy source for allowing cells to take on all kinds of different types of characteristics. So mitochondria are bacterial derived, and their energy metabolism is absolutely linked to all kinds of things happening within the cell. And that's a really fascinating area of cell biology that I think we're still just at the tip of the iceberg in terms of understanding. The nucleus, of course, is where DNA replication and storage is occurring. Peroxisomes, many of you, took me a while to really understand peroxisomes, but they are a major detoxification system, and they also make a lot of different unusual types of lipids, including precursors for lipids that myelinate all of your neurons in your brain. And the lipid droplet is a fat storage organelle that turns out to be extremely important for allowing mitochondria to drive energy production when you are fasting or when cells are starved. Each of these organelles, if the components of them are mutated or disturbed, will underlie many medical conditions. So basically, all of the medical conditions that you see to the right here, many of them are generated as a consequence of mutations in proteins in these different organelles that are going awry, from Parkinson's disease to many neurodegenerative diseases, and bone disorders are involved in misfolding of proteins in the ER, the inability of these proteins to effectively enter the secretory pathway, et cetera. And in the case of peroxisomes, mutations in their biogenesis give rise to terrible neurodegenerative disorder, Zellweger syndrome. And lipid droplets are involved in many lipid inflammation and immune disorders. So if we're going to be able to, at any point, really understand these diseases, we need to understand how these organelles are actually operating. And that's been the goal of my lab for many, many years, is trying to understand how all of these organelles coordinate with each other to give rise to cell homeostasis and the health of an organism. Now, over the course of almost 30 years, There's been tremendous developments in imaging technology that are expanding our understanding of these diseases. And this is just a list of some of these that I've been fortunate enough to be able to be involved in. But basically, they range from diffraction limited imaging to photoactivation to super resolution imaging at different scales, and ultimately to new electron microscopy approaches that allow you to correlate light and whole cell film, whole cell focused ion beam scanning EM. Now, for many of you who are interested in trying to understand cellular behavior, the first choice that you have to make is, am I going to use electron microscopy or am I going to use light microscopy? And I think that this is a choice that doesn't need to be made. I think you need to combine these approaches whenever you can. They offer very different perspectives on how cells are organized. The light microscopy, which I'm going to be talking about in my second talk, has the advantage of giving you global context, protein specificity, because you can introduce specific fluorescently tagged proteins, but it also allows you to gain incredible insight into the dynamism of a particular organelle, as shown in the movie that is illustrated here, where we're looking at the dynamism of the endoplasmic reticulum. Now, electron microscopy, by contrast, gives you exquisite ultrastructure. And if you're using certain types of microscopy, like I'm going to be talking about in a second, focused ion beam scanning electron microscopy, you can get isotropic resolution across large volumes. So let's focus in on this new type of volumetric electron microscopy called focused ion beam scanning electron microscopy. Basically, the approach is to embed your specimen, be it a cell or a tissue, in a block. And on one face of that block, you use a scanning electron microscope to image at high resolution. And then you use a focused ion beam perpendicular to that to shave off a different layer in the block for you to then image again with scanning electron microscopy. And if you do this over and over again, you can essentially section your way through the entire tissue, as shown here, where we're sectioning through an entire HeLa cell at 4 nanometer voxels, or 4 nanometer slices, if you will. This technology was made available at Janelia by Harold Hess by really tweaking existing FibSim microscopes so that they can operate over long periods of time. To section through this entire HeLa cell with these very sort of narrow slices, if you will, took almost 2 weeks for that machine to be able to work nonstop. A lot of commercial machines will work for a day or a few hours, and then you'll get a glitch. So it's critical to have machines that can continuously engage in this cycle of milling and scanning EM if you're going to be able to get through large volumes. So what do you do when you get this kind of information? It's a tremendous amount of information. Well, what we were interested in was looking at all of the different organelles. So here's a 1 cubic micron section, we just take 1 little cubic micron area out of that cell that you just saw, and by hand, postdoc Aubrey Weigel in the lab segmented out all of these different organelles, including the Golgi apparatus in red, endosomes in purple. You can see lysosomes, peroxisomes, etc. Now, this took Aubrey about 7 to 10 days to do this. The whole cell is 7,000 times this size. So if if we want to be able to do the whole cell, which ideally we would like to, because this is just one small part of the cell where there's this right near the centrosome, it would take 50 years for a single person to do that. So this is where we started thinking hard at Janelia about how we could potentially accomplish this. And what emerged is what we're calling cell organelle segmentation electron microscopy, or COSEM, which is a project team where we combine this EM data acquisition with people who can do machine learning to try to learn from manually segmented images, how to allow a computer to be able to do this automatically through a whole cell. And then the other part of the team are cell biologists like myself who then interrogate this data. So this is the pipeline for how one goes about collecting and interrogating the system. We have data pre-processing, which is basically just making sure that the data is all aligned properly. And then we have annotators who go in and do the hard job of annotating sub pieces of the cell that we're interested in being able to segment through automatically. And then those small little annotated parts of the cells are handed over to machine learning computationalists who then train the computer to do this automatically. Once that's done, it's handed back to the annotators who then evaluate how well the computer has done in that. And this is a sort of you go back and forth several times to make sure that the machine is learning properly and doing it properly. And then once that's happened, we have people who are involved in data post-processing and data analysis, and then ultimately data sharing, because this data is too big for just one group, one person to interrogate. Basically, this needs to be put out to the entire world because it's incredible amounts of information that we've only been able to sort of analyze at very small subsets of. So let's look at this process. Here's a FIBS. This is the sort of first step where here's all the data that's being sort of assessed and aligned. All of these individual cubic blocks are areas where our annotators went in and manually annotated that region. Here's just an example of some of the organelles that were annotated in order to do that. And then those annotations are given to the computationalists who then start training their computers to see if they can do it manually. And so here's an example where they're doing Golgi predictions, mitochondria predictions, endosome predictions, vesicle predictions, and then ultimately nucleus predictions. But basically, we've been able to predict up to about 30 different types of structures within the cell using this approach. We've done this for 4 different cells, 2 HeLa cells, a macrophage, and a Jurkat. This is just an example of a Golgi apparatus in this HeLa cell. This is the full Golgi apparatus in that entire cell. I just want to draw your attention to the contrast to the sort of image that you get of a Golgi in a textbook, which is just sort of a small little stack of pancakes. In fact, the Golgi is a very, very elaborate structure whose stacked cisternae are fully interconnected with each other and very elaborate, extends many, many microns through the cell. It was sort of mind-blowing to see this. From this data, we can begin calculating percentages, volume of different organelles relative to each other in individual cells and comparing them between the different cells. For instance, you can see from this, the Jurkat cell has much bigger nucleus per volume compared to these other cells. It also has a much smaller fraction of peripheral ER relative to, for instance, the macrophage. All of these pieces of information we're trying to now make sense of in terms of how this relates to what these cells are actually doing physiologically. This is just an example of some of the other types of unusual information that you can get from these data sets. Here's just a single microtubule. Each of those color spots on that microtubule represent different organelles that are that are touching that microtubule. Again, underscoring just how complex the organization of cells are. We know that organelles move along microtubules. Well, we've got 5 different organelles touching that single microtubule. So how do you orchestrate all of this? This is the endoplasmic reticulum towards the center of the cell. You can see it's got many different shapes and morphologies associated with it, including being planar in some areas, reticular in other areas. The planar areas frequently are abutted to mitochondria at what we're calling mitochondrial contact sites. So with that, I want to stop because I'm sure I'm running out of time. But I just want to emphasize that using these large volumetric electron microscopy data sets is really just opening up all kinds of possibilities for deeper understanding into how organelles are organized within a cell, how organelles are contacting each other, and potentially communicating with each other in ways that are crucial for setting up different types of cell identities and behaviors within cells. Thanks.
Thanks. That was honestly spectacular to see the sort of the resolution. You can see how organelles touch microtubules. I was, wow. Okay, but I am the chair, so I am not going to ask questions. So I already saw a question from Peter Zelstra, who's in the panel. Do you want to ask the first question? Yeah, we don't hear you. Still not. Okay, then I'm going to, Chase, can you read one of the first questions from the—
Yeah, I'll just read out Peter's question because he actually was kind enough to type it in. How long does the imaging take compared to the data analysis and interpretation?
So to image that HeLa cell took 2 weeks. If you're going to image like a big block of brain, which is what actually people are doing now at Jamelia, we're talking months. So it's really important to get these microscopes really set up so they run smoothly and routinely. Once you have the pipeline for analysis, the analysis is much, much faster. I mean, within just a few hours, you can analyze, once you have that program, and the computer knows what a lysosome looks like, what a mitochondria looks like, it can happen very, very fast. Within a day, you can see all of these organelles. Now, the first run through, of course, where we're working to teach the computer what is a lysosome, what is a mitochondria, that takes back and forth. But once you have the algorithms laid out, it's very, very fast. Now, one of the things that we're working on right now is, I showed you 4 different cell types where the same algorithms could be used to analyze each of the data sets. We don't know how well it's going to work in cells that we find in tissue, for instance, where things could be the labeling intensity of these membranes that we're using as a way to identify the different organelles can vary. And also the organelles look really different in different tissue cells, in cells within a tissue as opposed to cells that are on a cover slip. So that's where we're sort of moving right now, is sort of challenging the system for how well we're going to be able to use it. And it's not a worry. basically we'll just have to retrain the computers to be able to do that.
Yeah, this relates to a question that was in the chat also by Yanni Hariu, who asks, do you need to train a new machine learning model for each cell type, or can one model be applied to different cells?
Right, so the one model applied to all those 4 cells that I showed. Right now, we're testing the extent to which it could be used. For instance, if we had a tissue section from a brain, for instance, at being able to identify ER in a neuron versus an astrocyte, or identify endosomal structures in these different contexts. So we're still at a very early stage. I should say that this work is now in press and is already on bioRxiv. The data set can be downloaded by anybody, as well as the information for interrogating these organelles using the algorithms that we've developed. Our belief is that this is not something that an individual lab can possibly deal with other than focusing on like one small little section. So it's got to, with this kind of data, it's got to be open to everyone to be able to have the ability to mine, because there's too much information in it that's just really mind-blowing. Every time we look at these data sets, it's like, it's, you know, we see things that I would never have anticipated.
Yeah, it's overwhelming, yeah. Kristen, do you have a question?
Hello, thanks Jennifer for the talk. Yeah, it goes actually in the same direction. I was first also wondering how transferable is the machine learning algorithm among cell types, but that you already answered. So I was wondering if now someone were to take their own FibSen data on their own machine, and I mean, they will probably never be able to take the whole cell like you managed to do with the Janelia team, but if they take their own little section of the same cell type, do you think they could use your training data to analyze that? Did you check how different?
Machine learning, yeah.
Yeah.
That's a good question. Definitely it's worth a try. Right, I don't think we've done that. I can ask the team whether they would be interested in doing that. I mean,
because I'm just asking, I'm coming more from the light microscopy part, but I'm also looking at label-free imaging and using similar kinds of segmentation approaches. And there I know that it matters a lot. Like if you already change a little bit the objective, then your training data, you need new training data.
The problem is going to be, the FibSen section, you know, the individual sections I'm not worried so much about, but because once you know how whether you're doing 8 nanometer milling versus 4, you know, that can be taken into account. What I worry more about is how the staining, basically the way this, these are cryo-frozen samples, which is one really critical thing, that are then they are post-processed using an osmium and uranium uranyl acetate sort of type of fixative staining, if you will. And that makes a huge difference in terms of how densely labeled the individual membranes are, which can throw off the computational analysis. You know, for instance,
I understand. So
if it's not labeled strong enough, you can't, the computer can't see it, won't be able to pick it up. And you'll get all kinds of false calls on that.
Okay.
I think it's very important. You're going to have whoever. I mean, I think for sure there will be people who have these microscopes. It's not that hard. Well, I shouldn't say it's not that hard, but basically, I know Harold Hess and his team at Janelia are willing to interact with people who have these machines to work with them to try tweak the machine so that they can run smoothly over several weeks without breaking down or stopping and having to restart. And every time that happens, things get completely misaligned. And that's a real problem if you're trying to section through the whole cell. But my understanding is that working among these these groups of physicists, that more of these systems will become online as we move forward. So then the question then becomes analyzing this data using these computational approaches. And I think if you have one of these machines and you are collecting this sort of data, you're going to have annotators, people who will be able to, you can't, actually, some people will just send off the data to a commercial outfit that will then segment for them. And that can do, that works at some level, and there may be more commercial outfits that emerge with time as people get more and more excited with this technology. But if you want to do it in-house, you really need annotators, which can just be 2 or 3 people. But these are people who are going to really make sure that they can provide the ground truth for any kind of computational learning by the computer, but they can also make sure that the images are aligned properly, and once any segmentation that has been done is done properly.
Okay, I want to, I find a similar discussion. I want to ask one question that I got in the chat, and then we have to move on, but I thought it was nice because Rafael Lira writes, beautiful, mesmerizing talk, thank you. Question, in addition to the Golgi shape you showed, was there any other non-expected finding from these extremely high resolution images you acquired that surprised you?
So if you attend my talk in about an hour, I'll show you.
That's a nice break.
Actually, yeah, the secretory pathway. I'm going to show you some very interesting morphology of the ER exit sites and ER to Golgi transport intermediates that we would have never anticipated.
Right. Okay, so let's thank Jennifer again very, very much. And then
So I'll stop sharing.
Yeah, you stop sharing. So then, and we'll see you back in half an hour. So the next speaker of the session is going to be Maaike Hansen. And Maaike Hansen is actually recently, basically like the last years, started as a tenure tracker in Nijmegen University. And she combines biochemistry and biophysics to look at basically robustness and how this emerges from the complexity of life. Yeah, so maybe I don't want to spend too much time introducing her. I'm very much looking forward to hear what you have been doing and what you're going to do. So go ahead.
Great, thank you very much for the nice introduction, and thanks for inviting me today. Just quick check, you can see my screen okay? Yeah, okay. So yeah, I'm going to spend the next 15 minutes or so talking to you about how cellular modulation of gene expression can be achieved. And I'm going to use HIV as a model system for that.
It's not yet full screen. I don't know whether you mind, but...
Okay, sorry. Better?
No, maybe it doesn't matter. We see still your slides, but...
You just need to go into PowerPoint mode.
Yeah.
Still not?
No.
Okay, hang on. I'll just try starting again. Now?
Yeah, that works.
Okay, great. Thank you. Let's just see. Yeah. So first of all, I want to talk a little bit about gene expression variability and the context in which gene expression variability can actually be functionally relevant. For example, if we have sort of 2 proteins that somehow antagonistically regulate a certain function, then we can imagine that depending on the levels of both of these proteins within a cell at any given moment in time, that these cells might have very different functionally relevant states. And that means that if you have fluctuations of both proteins over time, that we can call protein noise or gene expression variability, that the actual function of the cell could also fluctuate over time. So the cell might make transient decisions about entering different functionally relevant states. So examples of these have been shown in bacteria, mouse embryonic stem cells, C. elegans, where a high amount of protein noise can generate cellular diversity. So just have different levels of protein expression in different genetically identical cells that result in phenotypically different cells. This protein noise can then also generate scenarios which are both beneficial or detrimental to the cells. So in other words, noise or gene expression variability can be selected for and against. Classic examples of where protein noise has been selected for is cellular bed hedging like strategies, where, for example, when these bacteria cells, or it's also been shown in cancer cells, are exposed to drugs, the fluctuations in mRNA and protein levels can allow for cells to enter drug tolerant states in a transient way, which means that if down here you're looking at the image of bacterial cells that through a certain amount of time or at a certain time point are treated with ampicillin, and some bacterial colonies actually can tolerate that drug treatment. Importantly, if that ampicillin is flushed out and then retreated, they might die, which means that this is not genetic, it is transient. On the other hand, this variability, this noise, is of course a consequence of sort of biology at a single cell level, meaning that it's not always beneficial. So certain genes, like housekeeping genes, have or show markers that they've actually evolved to select against this noise. So they want to reduce the amount of variability because it is very important for them to be expressed stably over time. Another very, very elegant, very beautiful example is in Drosophila development, where it's actually been shown that multiple nuclei can share the same cytoplasm, so that the transcriptional bursting that happens and generates this mRNA and protein noise is dampened globally throughout Drosophila development. So that raises the question, we now have scenarios where it's beneficial, there are scenarios where it's detrimental, but what allows some sort of temporal switch between a high noise scenario and a low noise scenario? So for that, we can use HIV as a model system. The reason HIV is so nice is because the output is binary and it's quite easy to read. So when HIV infects a cell, it integrates into the host cell genome, and then one of two things can happen. Either enough mRNA and protein can be expressed so that the virus undergoes active viral replication, meaning it produces new virions, it kills its host, and then it spreads. Or when it integrates, that threshold is not met. The virus goes transcriptionally quiescent, and it contributes to the latent viral population. So importantly, from a therapeutic standpoint, we can only target viruses undergoing active viral replication with antiretroviral therapy with current strategies. So that means that from the virus's perspective, it's beneficial to sort of lock both of these phenotypes into place and allow them to persist over time. So there are these two phases where a high amount of variability is beneficial for HIV to explore its different fate options. But then the virus wants to stabilize its fate, to commit to one of the fates so that it can be locked in place over time. So how does HIV achieve this switch? And it turns out there's sort of three main, or we can narrow it down to three important features. The first is that HIV mRNA needs to be produced in a cascade. So if you look here on the right, what you have is you have the HIV genome that's integrated into the host cell, and then you have transcription of a full-length transcript that is unspliced transcript. And then you have a splicing event, which is essentially an mRNA processing event where a chunk of RNA is cut out. And that generates the singly spliced transcript class. And then there's another processing step that generates the multiply spliced transcript class. Then the second thing that's important is that that there's a positive feedback that actually drives phase exploration. This positive feedback is mediated by this protein called TAT. It shuttles back into the nucleus and transactivates the viral promoter, generating this positive feedback that drives these large fluctuations in HIV protein expression, allowing HIV to explore these two fate options. And then the last thing that's important is negative feedback that is mediated by REV, which is another HIV protein. This negative feedback, again, the protein shuttles back into the nucleus and binds the unspliced and singly spliced classes, exports them into the cytoplasm, and that's how this negative feedback is generated. And now you can actually also see why it's important that this is produced as a cascade, because if there weren't this cascade or this precursor product relationship, this negative feedback just couldn't occur. So it's this negative feedback that stabilizes the commitment. So now I'm going to talk a little bit about the data that supports this. And the first is that we first want to show that the HIV mRNA is actually produced as a cascade. And we can do that using single molecule RNA fish that targets these specific transcripts with different colors. And then we can treat cells in two phases, different time periods, where we first have a TNF induction phase. And then after that, more importantly, we have an actinomycin D transcriptional shutdown phase. What that actinomycin D, it's a small molecule inhibitor of transcription, which means that when we don't have production of new transcripts, we should be able to see what happens after transcription occurs. So for the unspliced RNA, you can see that over time, these are representative cells, but here's the quantification on the right. And you can see that in red, once we stop transcription, as expected, the unspliced transcript just decreases over time. The singly spliced transcript instead, even though transcription is shut down, it increases and then decreases. And the multiply spliced class just increases over the course of the 40 or 50 minutes that we treat with actinomycin D. Showing this clear progression from unspliced to singly spliced to multiply spliced transcripts, and that mRNA HIV mRNA is indeed produces cascade. So the second thing we want to do is actually look at protein production in HIV. And what we can do there is we can use two fluorescent proteins as reporters. One is GFP, which is a reporter of the multiply spliced product, and then mCherry, which is a reporter of nuclear export. Then we can do microscopy of cells that are expressing these two reporters within the same cell. And on the left, you can see that GFP quantification over time, where you have an initial increase, and after about 5 hours, you have reached a maximum, and then there's a decrease, and after about 20 hours, you reach a steady state. However, the unspliced reporter, there's a delay. After about 5 hours, you start seeing a gradual increase over time. And this delay corresponds to the initial 5 hours of GFP production. So in other words, once you start seeing the export of the unspliced precursor, that is when you see the decrease in GFP production of the multiply spliced product. So you have this overshoot that is a classic hallmark of a negative feedback that corresponds to the export of the precursor into the cytoplasm. Now we can see at a protein level. But this doesn't necessarily get at, okay, what exactly does the feedback do and is it really there? So what we can try and do is genetically enhance these splicing events by increasing the efficiency. So we effectively get rid of the negative feedback, and we can compare this mutant, which is the positive feedback only, with both the positive and the negative feedback. And we can see what happens. And here is the quantification of the same time-lapse images. And you can see on the left is the positive feedback only. So this is the mutant, and on the right is both the positive and the negative feedback. And three things become quite immediately apparent. The first is we have gotten rid of the negative feedback because the mean expression level with a positive feedback only is higher. The second is by getting rid of this negative feedback, we've also gotten rid of this overshoot, which again is a hallmark of the presence of a negative feedback. And the third is by getting rid of this negative feedback, the noise in expression of HIV increases drastically. So in other words, the presence of the negative feedback somehow seems to attenuate these stochastic fluctuations, or these gene expression variability caused by HIV. So we can quantify the noise and the mean for both the mutant with the positive feedback and the wild type, and we can actually here it's makes sense. It's pretty intuitive. Over time, you have an increase in both mean and in noise because there's only the positive feedback. However, if we look at the positive feedback and the negative feedback, you see these two distinct phases of the noise and mean trajectory, where you first have an increase, which corresponds to the positive feedback. Then there seems to be some sort of competition between the positive and negative feedback, where you have a plateau of both the noise and the mean sort of stays the same. And then you start seeing a decrease in both mean and noise, which is when the negative feedback takes over and presumably stabilizes commitment. So, of course, now we've only shown that there is a decrease in noise, but what's the functional advantage of this? So we can repurpose this experiment a little bit, and what we can do now is we stimulate all our cells to force them into the active state. And then we wash out TNF, which is the small molecule that stimulates these cells. And then we can ask, comparing the positive feedback versus the positive and negative feedback, which of the two scenarios is more stable in the active state? So we can watch over time, over the course of 58 hours, what these distributions do. And you can see that after 58 hours, the wild-type HIV that has both the positive and the negative feedback remains in this actively transcribing and translating state longer or more stably than the mutant that only has the positive feedback. So one final question is, does this hold also for the latent choice, right? So we have a binary or two fates. Experimentally, this is a little more difficult to get at, which I'm happy to discuss later. But what we can do is we can run some stochastic simulations to get at this. So we can model essentially this schematic that I'm showing you here that importantly has both the positive and the negative feedback. And we can run these simulations, and we can select only those trajectories that are off, so that have chosen this latent fate. And then it's known that HIV randomly reactivates, generating these so-called blips. And we can ask, if a blip happens, what's the probability that our three scenarios somehow manage to mitigate that blip and push it back down into the latent state? And we've done this in the absence of the feedback, so no rev, wild type levels of rev, so intermediate levels of rev, and a high amount of rev. And as you increase the amount of rev, the probability that there's a decrease in these stochastic reactivations increases. In other words, the presence of rev also stabilizes the latent state. So I hope that I've shown you the different data that supports the fact that the way that HIV at least modulates gene expression variability is by requiring a cascade production, and it has both a positive and a negative feedback that allows for a temporal switch between sort of this exploration and this commitment. So we can ask, can we find these motifs in other systems, right? HIV, it's a virus and quite an easy binary, as I said, model system to study, but we want to know how pervasive this is. So we can go into literature and we can find some of these architectures, and through, again, simulations, we can figure out that they do reduce noise. But sort of ending this on a little bit more of an open question side, it's of course very hard to show that just because a feedback can modulate noise, does that also mean that it's a functional advantage of modulating noise? So something that I at least always need to remind myself of is because I love sort of the single molecule stochastic nature of cells, but just because something is fluctuating or random or noisy doesn't necessarily mean that it's functionally relevant. And one of the problems is that the fact that noise is a problem, we can quite easily pick up on features of those, because to suppress noise or variability, it's quite costly for a cell to do. So the mechanisms that are known are just generally not very efficient. So we can use that argument quite strongly and say, well, in these cases, noise has been suppressed and is therefore unwanted. However, for noise to be beneficial, it's hard to separate between it actually being beneficial and used by the cells or simply not detrimental, and that's why not filtered out. So that's just something to keep in mind. And the other thing is a lot of perturbations that one does to try and get at noise effects also inherently change the mean. So it's very difficult to separate out sort of functional effects of noise versus mean effects. And then just leaving you with sort of one final open question, the question of what exactly is noise, I think can be separated into two parts. One is, the simplest random event in a cell is the random collision of molecules. So inherently, everything in a cell needs to be non-deterministic and stochastic. That's one extreme. Or the other is, well, if we can measure every single aspect of a cell, then we can assign everything, right? Then it's not actually noise, it's just stuff that is undiscovered or unassigned. So does that mean that if we discover or assign all the variability in a cell, that we can find the simplest stochastic event? And is that a cause or not? That's sort of the question I want to leave you with. So with that, I of course need to thank some people. So what I showed was work that was primarily done throughout my postdoc. So that was with Lior Weinberger, who's a phenomenal mentor. And then some additional work by some group members, especially Oscar and Benjamin. And then, yeah, funding collaborators and you for your attention. Happy to take any questions.
Yeah, thank you very much. Yes, so any of the panel members would like to start with a question, and for the people who are attending, you can put your questions in the chat. Yes, Tom?
Yeah, I could ask a question. Yeah, it's great talk, beautiful data as well. So I was curious, in the end, your story sounded like, I guess for the purpose of the HIV's biology, I guess the idea is that it's really trying to suppress noise. Is that the way you interpret this? That it's a noise suppression mechanism at work?
Yeah, so the negative feedback is. The positive is a noise enhancing mechanism. And it's just the time period in which each of these dominates is different. So early stages, it's noise enhancing, and later it's noise suppressing.
Yeah, and for the noise enhancing phase, is there a sense of, whether there is a biological relevance to the enhancement?
Yeah, because, well, it's split, but the positive feedback actually allows for both the active and the latent population to establish. Mm-hmm. And both of these populations on the long-term perspective from the virus are beneficial for, like, if it would only actively replicate, at some point we could treat all of the actively replicating HIV and it wouldn't persist over longer time periods. And it's that latent population that's a problem for treatment.
I see. So it enhances this commitment step, I guess, that you talked about.
Yeah, so the positive feedback actually causes bimodality. So it causes an off population that's the latent and an on population that's the active. And the negative feedback allows the on population to stay on and the off population to stay off. So there's no constant switching over time.
Okay.
Yeah. Okay.
Thank you.
Okay, thank you. Maybe I have a question, Maaike, because it especially, of course, was interesting your statement, or like, I guess, something that keeps you busy. Like, is it relevant? Which seems, I guess we all struggle with that sometimes. So it's very.
Is it just fun?
Yeah, exactly. But so because you're stating like it's you don't know whether it's just there or it's beneficial. But I think at least in yeast, what people have done is they have characterized very many promoters, and then you know what the mean is and the standard deviation and your noise level, and then you can start to swap around promoters. That at least, I guess, for yeast, that's a way of addressing that problem, right?
Yeah. So it's a very elegant way of addressing the problem, and it can be done. It's just difficult also to because different promoter architectures have higher or lower noise, but that higher or lower noise is also associated with different levels of different mean levels. So then you need to compensate. If your goal is to keep the mean the same, you need to compensate and change your degradation rate of the transcript or protein, which in itself might also have an effect. So I'm not saying, I think that those studies are convincing, just it's very hard to detangle the mean and noise effect. And it's just something that is good for all of us to keep in mind.
I didn't really know this. I thought, but maybe I never gave this a lot of thought. So what I thought was probably too easy was there's just a lot of different promoters in yeast. And there's just a lot of different, and that mean is not very strongly correlated with expression level. There's probably some correlation, but if I have a whole set of 1,000 promoters, I can just pick the one that happens to be sort of the right, because degradation levels are also measured, right? So I thought the library is large enough so that I basically, although there's this correlation, I can still pick the one that I want that has a certain sort of, that has the right expression level and the right mean, although there's some correlation. And do you now say that that's not really true, that actually there's not so much, that there's only a few, in essence, only a few promoters?
No, okay, I'm looking at it a little more sort of mathematical. If you look at the duty cycle and promoters are on versus off, let's say to get the same mean, you need a promoter to be on 50% of the time. And that can either happen between it being on and off very fast, you can get the same mean depending on the promoter architecture. But I think if you start switching around just promoters, you're looking confused.
I really thought of something. I never thought about it this way. I now realize you're right. I didn't think very deeply about how this. I thought that you have much more control, but it's right. So either way, on and off, and it's the time you're on. So I thought you can do things faster or slower, and that there are more different ways.
No, you can't just slow things down, right? You need to keep so assuming everything else downstream stays the same, degradation, exports, protein production, that stays the same. The only way you can keep the mean the same is if your total, your fraction of time that the promoter's on remains the same. So you can change, you can modulate that by changing the time intervals in which they're on and off. So you can toggle it faster or slower.
Okay,
so that you can change.
Yes, I understand. But now, okay, what about the following? Yeah. I thought about it. I teach about this actually, and I thought that the main cause of like fluctuation in protein levels is actually from burst in gene expression rather than sort of fluctuations in actual numbers of on or off states. Like, so that the burst size is also something you can control.
Yeah. So actually, burst size is the off rate, so the time that the promoter's on, over the number of transcripts that are produced per burst. So just mathematically, you can increase your burst size by keeping your promoter on longer or producing more transcript in that same time period that the promoter's on.
Yeah, exactly. I see now. Yeah, so you do really only have a few different, you have only very few knobs. I understand now. Yeah, exactly. So it's not, so you don't, in the end, okay, I understand better now what you're saying. So they are coupled. They're not fully independent. And biology doesn't have smart ways to control this in such a way that you still get a huge diversity in expression level and mean level and noise levels.
Yeah. So they're dependent, or a lot of things you change will change, or it's hard and laborious to find ways to change one independently of the other.
Okay, I got taken a little bit away because I did.
But I just full disclosure, I've never worked with yeast myself, so I'm not aware or like fully. I know eukaryotes. I know a little bit about bacteria, or higher eukaryotes. Sorry, don't want to offend any yeast biologists. So it could very well be that it's way. I mean, it probably is way easier in yeast than in higher eukaryotes. But don't want to tread on any toes there.
No, sure. I mean, I won't say I was carried away because I'm a chair, and I actually just noticed that Peter also has a question. You want to ask a question, Peter?
Yes, I hope you can hear me now.
Yeah.
Yeah, great. So very nice talk, Mike. I had a question about the results you showed where you averaged the data over many cells. So you had the mCherry and the GFP expression there. And I was wondering, there seems to be a big difference from cell to cell as well. And is the origin of that the same underlying mechanism of this noisy protein expression, or is there an additional mechanism that causes intercellular heterogeneity?
So they both contribute. So the first thing I should say is that the quantification, we sort of in silico adjusted the time frame. So you could see in the video that some cells hadn't quite started expressing yet, and we just aligned them so that they all start expressing at the same time. And this might get into your question. There are known factors that are we call them extrinsic, that contribute to this from one cell to another. Differences like cell volume, cell cycle state, things like that. The classic sort of separation is intrinsic and extrinsic, where intrinsic causes ergodic fluctuations and extrinsic can cause overall expression of one cell to be higher than the other cell. I think so that means that if more, sorry.
Sorry. So that means if you divide your cells into subclasses of different volumes or so, then you would get narrower distributions because you narrow down on these extrinsic fluctuations. Is that correct, or?
In our analysis, no, because you can actually, so it's called, well, we call it extrinsic noise filter. So you can actually see, if you look at just one cell over time, it will fluctuate, right? And that behavior is presumably ergodic, meaning that the full fluctuation, or if you sum all those time points, it should make a distribution that is equivalent to your intrinsic noise. But you can also calculate, okay, what are my differences caused by volume or other factors? And that's not what we look at, because we're interested in these ergodic fluctuations and not these cell-to-cell differences caused by external factors.
Okay, I understand. Yeah, okay, thanks a lot.
I think you're muted.
Thank you. Well, that happened at least once. So I think we have time for one more question. I saw that Hilke, you raise your hands.
Yeah. So thank you for your nice talk. This gives me lots of thought for thinking. Regarding this kind of binary behavior of negative and positive feedback, that if you presume that this doesn't change the mean but only the noise levels, do you think that adaptation of that negative versus positive feedback mechanism also is basically adapted towards the biological kind of system a protein functions? So I can understand, like, for example, some biological function need to be need to happen through bursts of proteins, whereas others just need to have a constant kind of replenishment of proteins itself. So there is like a difference how a process works, positive versus like the positive feedback versus the negative feedback.
Yeah, so I'm going to reinterpret your question just a little bit and reformulate it maybe. So the question we're actually now trying to ask is a positive feedback always noise generating? Or can we find positive feedback that act differently, that aren't transcriptional, that are also noise attenuating? right? So that you can actually find or separate out the space or the functional space of mean and noise into 4 different directions. The, I think, more direct answer to your question is actually these feedback mechanisms, they are stronger or less strong at noise attenuating and amplifying depending on the promoter architecture. So this actually gets a little bit back to Liedewijs or our discussion earlier. Depending on if the promoter is always on or if the promoter is more bursty, these feedback can be more noise amplifying or less, or noise attenuating or less. So I definitely think there's a lot of fun questions to ask here to say, okay, is a protein that has a certain function that requires this sort of promoter architecture, is it also found more often with a certain feedback architecture?
Yeah, exactly. And I was wondering then if that only is, we can only work that out regarding the promoter architecture, or is this also seen in the functioning of the protein itself? So do you see certain types of proteins which have...
So honestly, I haven't looked, just because my curiosity is more on the promoter and transcription side. I think it would be fascinating to find that I don't know, maybe splicing proteins themselves are regulated in a certain way. Like if you have these sort of classes of proteins, or proteins that carry out, I mean, you know that, or it is known that stress response genes are more noisy, but I don't know what sort of feedback mechanisms are found within these proteins. But I think, yeah, that would be a really fun question.
Yeah, I'm thinking like cell cycle dependent.
Yeah.
Yes.
Okay, I'm going to going to intervene. I thank you very much, Mike. I think I really learned something. That's always very nice. So thanks for the good talk. Let's take 4 minutes and then go back at 16:40. We reconvene and we continue with Jennifer's tutorial. And then also, I think Chase is going to let everyone in that is still here to come for the tutorial. So everyone can enter this room now and we can have like a classroom setting. Yeah, that's it. So yeah, at 16:40 we continue. That was very nice, Mike. Oh.
I find it a little weird with the questions because you're only looking at the camera, and usually I try and figure out if the answer is like sparking some sort of understanding or not.
Right, yeah, it's very strange. It's also if you teach, right? This is a strange thing, but yeah, it was in We Should Talk once. It's very interesting. Yeah, nice stuff.
So your background is with yeast, or
yeah,
for the teaching that you said?
No, I do actually teach. We work with yeast, but we also, I mean, we actually look on polarity establishment. But we start to be interested in noise because we realize it has effects. So we sort of stumbled into noise. We work a lot with feedback, but I never thought about noise so much before. So from that perspective, we work with and yeast is just one of our models. So we do both in vitro constitutions, but also yeast. And I like about yeast because in that you have a lot of ways to perturb, so you have some control. So I was thinking about, actually, because we think of a role for yeast, I was thinking to take promoters, to swap promoters and see what happens if I increase the noise level. So I was very with this idea, but now I realize that it may be more complicated than that. But that's good to know. Better to know.
I think it's a fun approach. Some things to keep in mind.
No, that's exactly right. That's exactly right. It's only good to keep that in mind. Yeah. Okay, I noticed that people are starting to shift from the attendees to the panelists, so this is nice. Yeah, so it's about time. I would ask people, I mean, anyone who only if you feel comfortable, but you can switch on your microphone, your camera as well. We can feel like a little bit like we're in a classroom setting, and but just if you feel comfortable, of course. So yeah, I think there's no need to wait. So yeah, Jennifer, just share your screen and give us the follow-up on this. Apparently, what was it again? What we learned from the super.
Yeah, exactly. So just going back to where we were when I was talking, I mentioned all of these different organelles within eukaryotic cells, and the goal is to try to understand how these organelles are communicating with each other in different ways. And in my prior talk, I gave you some insights into some new electron microscopy approaches using focused ion beam scanning EM. Now I want to move to light microscopy and what it's sort of allowing us to understand. In particular, I'm going to be focusing on the endoplasmic reticulum, which you're seeing here in a Cas7 cell that's been labeled with an ER marker that I'm going to use frequently in subsequent slides called Sec61 beta. It distributes throughout the entire ER. What I want to draw your attention to is that the ER forms the nuclear envelope, and then it extends out to the periphery of the cell as an elaborate tubular reticular meshwork. It's a single compartment, so all of these membranes are connected to each other, which means if we photobleached a live cell expressing a molecule like Sec61 in a small area and continue to photobleach that area, we would ultimately photobleach every molecule within the ER, because these molecules are diffusing and essentially moving throughout this whole system. So this system is incredibly important to how proteins are being synthesized in cells. These are the proteins that make membranes that are secreted from cells. The ER also houses a large amount of calcium in the cell that can be released in different times and places. And the ER also is communicating with every other organelle within the cell through what we're calling contact sites. So I want to talk about three particular aspects of ER organization. One is the contact sites, where the ER has drawn close together with other organelles through tethering factors, and is communicating with these other organelles at those contact sites by either releasing calcium or other metabolites at that site, or undergoing direct lipid shuttling from ER to the other organelle, or vice versa. I'm also going to talk about, if there's time, ER matrices, as well as the ER exit sites, which I mentioned. We have some really exciting data related to their ultrastructure using cryocorrelative approaches. So I understand that people can interrupt at any particular time in this discussion, so feel free to do so. So let's start about these ER contact sites. We know they exist from early work we did a few years ago, where we expressed 6 different organelles simultaneously within cells, and you can see 3, 4, 5-way contacts between these different organelles when you reconstruct these. This is a 3D lattice light sheet movie series where we've done multispectral unmixing, so that you can see fluorescently tagged proteins associated with each of these organelles. This is just an example of some of these contacts, in particular a contact site that we're going to be focusing on in a couple of slides, which is the extent to which the ER is contacting mitochondria. To the left is a segmented mitochondrial system from the cell that you just saw. So basically the 3D lattice light sheet images allow us to segment out every single mitochondria. And we're looking at the dynamism of those mitochondria. To the right is where we've superimposed all of the regions of the ER that are sharing a pixel where mitochondria has been segmented. And what you can see is a large swath of the mitochondria, in fact, is in very close association over significant time periods with mitochondria. So two postdocs in the lab, Chris Abera and Johnny Nixon-Abel, were interested in tracking these molecules, in particular looking at proteins that might be involved in activity at these contact sites and how their single molecule motions might be impacted. So what you're looking at here is a freely moving molecule within the ER. We can do this using a halo-tagged KDEL containing protein that's been integrated into the ER lumen. So it's freely moving around the lumen of the ER, which you can, and the ER itself you can see in pink using an MM-rolled ER3 probe. So basically, unless molecules have particular, associations that anchor them to a particular place, we see them freely diffusing within the ER. Now we're going to look at a protein shown here, VAP-B, which we know is an ER mitochondrial tethering protein. In fact, it tethers the ER not only to mitochondria, but to other organelles as well. When we look at its trajectories using this single particle tracking analysis, what you can see is that many of these trajectories, in fact, if you look at this single trajectory down here in the bottom right panel, show free diffusion, and then suddenly you'll see them hovering, still undergoing motion, but very localized motion. We can see this for many places along its trajectory within the ER. If we then, yes.
So I was just curious. So in your ER, is the diffusion constant of these proteins, like according to their size or their predicted size, similar as in the cytoplasm, or is the ER, does it have similar sort of chemical properties, the inside of the ER, as the rest of the cytoplasm?
So, so work that we did many years ago using photobleaching revealed that a lot of these proteins, depending on whether you're a soluble protein or a membrane protein, you diffuse 10 times, you diffuse 10 times faster if you are a free soluble protein in the ER lumen compared to a membrane protein.
Right.
Most of the membrane proteins shared very similar diffusion coefficients because it was limited mainly by diffusion in the bilayer, and VAP shows that characteristic as well. So VAP is actually when it's freely diffusing in the ER, it's just diffusing along this bilayer, and that's very different than what we were just looking at, which is a molecule that was in this lumen, which actually diffuses faster than VAP-B. But the point that I want to focus on here is this change in its trajectory, where it's freely diffusing and then suddenly it comes up to something that changes its the extent of its motions. And if we do double labeling with a mitochondrial marker in red, in addition to our ER background, and look at where these trajectories are undergoing this type of hovering motion, we see that it is invariably at mitochondrial contact sites, as shown here. And to the left is just a diagram showing the protein on the mitochondria that the VAP-B is known to interact with from biochemical work. and that is this PTPI51. In fact, if we overexpress the PTPI51 in cells, we can end up with the ER just completely wrapping around mitochondria. But we were interested in looking in unperturbed cells at these contact sites. And to the left is just showing the ER as this white background, this reticular background. The mitochondria is this elongated structure here. In yellow is the outline that the yellow outlines the position of the mitochondria over this 60-second window of imaging. To the right, what we're plotting is the normalized localization probability of that beat over this 60-second time window. And what you can see is these hotspots for its localization. So the molecules are spending most of their time at these sites which we believe are the contact sites. And one reason for thinking that is that they have the very similar shapes and sizes to contact sites that we can see at the electron micrographic level. So here is a FibSim reconstruction to the left showing mitochondria, and here's the endoplasmic reticulum. This is the contact site shown in red here. If we flip this on its side, you can see the overall shape of that contact site. If we come down here, you can now look at contact sites that we're seeing in our single molecule analysis, and you can see that they have very similar shapes and sizes as what we're seeing at the EM level. This is another example of this, where we're looking at all of these different contact sites. These are the trajectories that allowed us to sort of define these sites. This is single molecule imaging. So what we're seeing is single molecule localization probability. So we're down to super resolution in terms of looking at these contact sites. Now, this kind of analysis has advantages over just electron microscopy because we're seeing a protein that forms these contact sites. And we can look at its diffusion coefficient as it moves in and out of these contact sites, as shown in this distribution plot, where each of these different spots represents a single molecule whose diffusion coefficient we have mapped. And as you can see, as you get closer to the center of this contact site, the diffusion coefficients drop and drop. And this is just plotted here, where we're looking at the diffusion coefficient across a line across that contact site, and how that correlates with the localization density of these molecules. So we're now considering various biophysical models for how these molecules might be being attracted and stabilized at these contact sites. Right now, we don't have a full characterization of it, but we think that this type of data offers a lot of possibilities in terms of really analyzing, the changes in affinity. I mean, do these molecules localize at these sites just through a sort of stochastic mechanism where as they get more and more clustered there, and these interactions continue with the mitochondria, there's no change in affinity per se, but it's just a clustering phenomenon versus some type of change of affinity as a consequence of the molecules the two membranes being closer together. We don't know at this point. But what we do know is that if we alter conditions of the cells, like here, where we're starving cells for 8 hours in a Hanks buffered salt solution, that these contact sites change. And we did this because we know from EM studies that the cells will change the extent to which these organelles are communicating with each other under starvation conditions. In particular, the ER and mitochondrial engage in more contact with each other to facilitate the mitochondrial's role during starvation. So mitochondria actually take charge of the cell, if you will, by producing energy under this type of starvation regime. And in order to do that, they need lipids and they need metabolites from the ER. And so what we can see is to the right, these contact sites are getting larger, and we're seeing changes in this diffusion coefficient profile of molecules that are distributing within these contact sites.
Jennifer, can I just quickly ask a question about how you actually estimate these effective diffusion coefficients? Are you essentially assuming it's free diffusion everywhere and then trying to do some kind of local estimator of the diffusion coefficient?
Yes, exactly. So we have the VAP-B and the PTPI51 are just one example of a contact site tether that links ER and mitochondrial activities. There are these other tethering components, and we're now aiming to tag these different molecules with our different fluorescent photoactivatable probes in order to look at their organization in these contact sites. For instance, is MFM1 and MF2 in the same contact sites that we're seeing VAP-B, or are they different contact sites? Right now, the field doesn't really know that because you cannot see these contact sites at the light level unless you do this type of single molecule tracking and probability distribution analysis. I now want to move to a slightly different feature of the ER, which I think is equally interesting to these contact sites, and that is the ER matrices.
Jennifer, may I have a
quick
question about the contact site still?
Yes.
I was wondering, looking at this data set, it seems to kind of hint towards that. I kind of have a chicken and an egg kind of question. Like, what influences the contact shape? Is it like the mitochondria shape who affects the ER, or is it the ER affecting the mitochondria? Regarding this data set you have here, it seems to hint towards that the mitochondria shape.
Absolutely. You're totally right. The mitochondrial shape is impacting absolutely basically the ER is just folding itself around the mitochondria and sort of flattening itself around particular domains of the mitochondria. And you can see that in this EM micrograph here. So here's the ER, it's just sort of laying itself, flattening itself onto the surface of that mitochondria. It's a really interesting point that you raise.
Cool data. Yeah, thank you.
So I could add one more question about,
sure.
It's an elementary question about this beautiful matrix, or I guess network of tubular structures that you see. I was wondering if it's known what gives it its structural stability or integrity, so that these really thin tubes are just really stable over long times. Are there any cytoskeletal sort of?
So that's what we're going to talk about right now.
Okay.
So these tubes we think are tubular membranes are a very fundamental feature of lots of membranes within the cell. Endosomes are tubular, lysosomes are tubular. I'm going to show ER to Golgi transport intermediates are tubular. So there's, it's a low energy state, if you will. But what's interesting about the ER is that these tubes come together into these three-way junctions. And that's thought to be mediated by particular proteins. But what I'm going to show is that in addition to these three-way junctions, you have more complex, very tight associations. And that we're interested in looking at using live cell imaging approaches and looking at the role of cytoskeleton. So this is a 3D section of we're just moving through the whole cell so that you can look at the entire endoplasmic reticulum through a whole cell. And And if you move out to the periphery, you start seeing these structures here where these three-way junctions seem to be sort of feeding into. And for many years, people thought these were just sheets of ER membranes out in the periphery. And in fact, lots of people had proposed particular roles of proteins involved in these sheets. But what we found several years ago using this technology, grazing incident turf, combined with structural illumination microscopy, the turf allows us to get a single plane of the ER at the bottom of the cell, and structural illumination microscopy gives us in 2 times improved XY resolution. What you can see is that the ER in the periphery here is made up of these three-way junctions and tubes, but the three-way junctions can come very close together into what we're calling matrices. These are not sheets of ER, flat sheets of ER. These are arrayed, clustered elements of three-way junctions. So, and you can we're just zooming up here to look at these things. I should say that this dynamism of the ER is basically reflecting sort of dynamic oscillations within the cytoplasm that's created by cytoskeletal behavior, because we can dramatically slow down these oscillations if we drop ATP levels. We were interested in what cytoskeletal system might be regulating this architecture of these matrices. And so Andy Moore just did some very quick correlations of the organization of the ER with microtubules on the one hand and vimentin, which is an intermediate filament, on the other. We chose microtubules because we already know that the ER can pull itself out on microtubules. And we chose vimentin because vimentin actually, if you look at it, has sort of a pattern that resembles the ER. So we basically did a ER radial distribution analysis. We spread the ER out on a single planar perspective. And then we do the same for the distribution of microtubules in that cell and the distribution of this intermediate filament protein, vimentin, in the cells. And what you can see is that the microtubules the radial intensity plot of the ER does not match microtubules very well, but it does match vimentin pretty well. And it doesn't, I should also say, we've done this for actin, and actin doesn't match this at all, because most of the actin is out at the periphery near the plasma membrane. So what is it? Where are there some correlations between vimentin's distribution and the ER? And what you can see with these yellow arrows is that areas where vimentin is being sort of clustered into what we're calling these knots are precisely areas where the ER has coalesced into these so-called matrices, which are just clustered three-way junctions of this membrane system. This is a movie where you can see the vimentin in white here is actually adhering to the surface of the ER. And as the vimentin forms these tight knots, it's coalescing into these tight knots, it draws with it the ER into these matrices. So we think that the dynamic behavior of vimentin, which is an intermediate filament that can adhere to the surface of the ER, is playing a role in restructuring the ER to form these matrices. And just as, here's another example. Here's vimentin in the far right. This is vimentin. You can see it's precisely overlaying the area of the ER labeled with this KDEL probe, where there are these tight matrices. And we can track the vimentin knots that we are associating with these ER matrices in time. And what you can see is that they all seem to be being drawn towards the nucleus, towards the center of the cell. And that makes sense because intermediate filaments are known to link the periphery to the nucleus. And as vermetin draws itself towards the nucleus via these contractile knots, it is surprisingly also drawing the endoplasmic reticulum towards the nucleus through this, we think, adherence between the vermetin, and these ER matrices. So what we're seeing here is just tracking of the ER matrices, and the tracks perfectly overlap where the tracks of vermetin are moving. So basically what we're seeing in these movies is that the formation of these vermetin filaments into these knots reorganizes the surface of the ER and drags it with it to the center of the cell. So how might this be regulated? How could the vermetin knots stabilize themselves on ER matrices? Well, we sort of opened our textbooks and started looking at what links intermediate filaments to ER. And the only thing that we could find is the linkage of intermediate filaments with this protein called Nesprin-3 and plectin, which are thought to anchor intermediate filaments to the nuclear envelope. So Nesprin-3 is synthesized in the ER, but diffuses onto the outer nuclear envelope where it can interact with these sun-tag proteins, but it also recruits plectin that binds to intermediate filaments. So we wanted to know, is it possible that this complex of Nesprin-3 and plectin in the ER, before it has reached the nuclear envelope, could be organizing these intermediate filaments and thereby not only organizing these intermediate filaments, but the plectin and Nesprin-3, if they can associate with ER, might essentially draw the connection between the intermediate filaments and the ER, because Nesprin-3 would be anchored in the ER. So this would be our ER protein that recruits a linker that then binds to vermetin to enable this association. So let's look at plectin first. So here's vermetin in the periphery of this cell. Here are the knots that we know are colocalized with the ER matrices. And we can see that this plectin, an antibody plectin, is colocalizing with vermetin. Here's the Pearson's correlation coefficient for that. If we rotate the anti-plectin image 90 degrees, we lose that correlation. This is just a zoom up of the plectin and vermetin colocalization. So yes, plectin does interact with these intermediate filaments out in the periphery of cells. What about Nesprin? Can we see Nesprin-3 in the ER? And when it is there, is it interacting with plectin intermediate filaments? And to do that, we first stained our cells for ER using our Sec61 marker. We then stained cells with vermetin, as shown here. And you can see that these two patterns very nicely correlate. The areas of the ER where there's a matrix correlate with where there's vermetin. Okay, so where's Nesprin-3? And you can see that Nesprin-3 is right at the ER matrix area where the vermetin knot is, as shown in this merged image and this zoom up, where here's the ER, here's the vermetin, and here's the Nesprin-3. So basically what that suggests is that, yes, indeed, there's this linkage between ER, the intermediate filament vermetin, and it's mediated by these two proteins, Nesprin-3 and plectin. So what are these ER matrices doing? If the cell has gone to this much trouble to link the ER to these intermediate filaments, what's the purpose of this? I mean, the idea is that somehow these intermediate filaments are restructuring the ER into these arrays of tight arrangement.
Can I ask you a question?
Sure.
We're
Yeah.
moving from the vimentin knots.
Yes.
So this vimentin knots, is it sort of, I find it quite interesting. Actually, I don't know so much about intermediate filaments. So is the idea that there may be myosin-like proteins that are then sort of condensing this vimentin, or how should I think about knot formation of this vimentin?
Right. So vimentin can, like actin, can undergo a sort of, vimentin, I mean, vimentin is really interesting in terms of a cytoskeletal filament. It can, well, it's definitely not using a motor to contract. It's possible that the ER,
nice, right.
that some sort of synergistic interaction between the ER's ability to these three-way junctions to diffuse and move around can stabilize interactions in the vimentin filaments themselves. So it's a synergistic interaction between these two systems. On the one hand, you have a highly dynamic array of ER and these three-way junctions. Vimentin starts locking onto it, and the two then self-organize into these structures, lead to the self-organization of vimentin into the structures.
The idea of motor is excluded somehow. People know that there are no motors.
Right. Yeah. Well, at least as far as I know, it's not like a myosin motor. There's no myosin motors that are associated with vimentin. There could be other components, but one possibility is it's the ER itself, and it's pliability that allows it to draw these structures inward. But one of the things that we found on these ER matrices was ribosomes, an enrichment of ribosomes that are involved in translation of membrane proteins and secretory proteins. And this is just a fib-sim reconstruction of the ER. So we just moved to our electron micrograph images, and we look at ribosomes, and we look for ER where we see these matrices. So this would be a matrix area. These are clustered three-way junctions. And you can see that these green structures here are ribosomes. We've highlighted all the ribosomes that are associated with ER, and they seem to be enriched in these three-way junctions or matrix areas. If we zoom in, you can see that the ribosomes actually are lined up in particular arrays, and these are polyribosomes, which are structures that form when a single mRNA that's in the process of translating is holding several ribosomes associated with each other. So this is telling us that there's active protein translation going on right there on that ER. Can we look at this in live cells? And Hee-Jin Choi, a postdoc in the lab, used SunTag ER labeling, looking at newly synthesized proteins using the SunTag system, where basically as soon as a protein is synthesized, it displays a target into which a fluorescent protein can bind, so that you can track where proteins that have just been synthesized are localized. And that's what you're seeing with this green signal here. In pink is an MS2 labeled mRNA that codes for the protein that we're looking at in SunTag. And the yellow trajectories are the movements of that mRNA. And you can see this is an active site of protein translation. And when we look at where that's happening, you can see it's at these three-way junctions here or in these matrices. So active membrane translation of mRNAs that are coding for membrane proteins is happening at these three-way junctions. Now, one of the really interesting observations that Hee-Jin made is that these translating proteins on the surface of the ER are doing it not only at these matrices, but in regions that are very closely opposed to lysosomes, which are shown here in red. And that was reminiscent, well, that made us think back on some other super resolution imaging we had done in collaboration with Dong Li, where we were imaging in our GI turf sim scope lysosomes in green and lysosomes in green, ER in purple, and the yellow string-like structures are microtubules. What you can see is the lysosomes are frequently trapped within these three-way junctions or matrices of the ER. So that led us to start thinking, okay, on the one hand, we know these three-way junctions and matrices seem to be hotspots for protein translation. On the other hand, there's lysosomes there. Is it possible that something is being released from the lysosome that facilitates local protein translation at these three-way junctions? And that something could be amino acids that are being released from a lysosome as a result of the lysosome's degradative activities. It could be mTOR, which is on the surface of the lysosome, or it could be pH changes. So to test that, Heejin basically blocked the lysosomal degradative activity with this protease cocktail. He increased the pH of lysosomes with chloroquine, or inhibited the mTOR activity of the lysosome with either rapamycin or taurin, one treatment. And what he found was that only inhibiting lysosomal proteolytic activity with this protease cocktail, or less significantly reducing and increasing lysosomal pH, led to a reduction in the translation of these of the mRNA that were associated with the ER for translation of these membrane proteins. So basically, to recap this data, what we're seeing is that ER matrices are forming and migrating inward through interactions with vimentin. The interactions, we don't really know who's driving who, but they're mediated by this Nesperin-3 protein that is a membrane protein of the ER, as well as plectin that binds to Nesperin-3, but also binds to vimentin. And importantly, we think that the cell is doing this because these ER matrices can trap organelles like lysosomes that can then facilitate protein translation at these sites because of their release of amino acids. And the amino acids are being used to build the proteins. So this is our current hypothesis for one of the functions that these ER matrices might provide to cells. So if you don't have any questions about that, I'm going to now move to the final part of this description, and that is the ER exit sites. So ER exit sites are distributed all over the surface of the ER. And they are the sites where newly synthesized proteins leave the ER to enter the secretory pathway. And that's illustrated in this movie here, where we're looking at a viral glycoprotein, VSVG, that has entered these ER exit sites and is about to then translocate via transport intermediates into the Golgi, which is the central structure here. The ER exit sites are known to be defined by coat proteins, COP2 in particular. It has been shown by Randy Sheckman to be critical for sorting of molecules to these ER exit sites. But once we follow these molecules as they move out of these ER exit sites, you can see they're moving on inward trajectories towards the Golgi apparatus. We know from prior studies that this is done through movement of these structures on microtubule tracks. So we wanted to get a closer look at this process. And to do that, we decided to combine light microscopy with FibSim reconstruction to get a fresh look at these ER exit sites at very high resolution throughout a whole cell. And so our reason for doing this, instead of just using transmission EM, which has already been used to interrogate these ER exit sites, is that the transmission EM work cannot do a whole cell unless you slice at fairly thick slices. And these large slices actually don't give you the kind of resolution in Z that you need to be able to get an isotropic view of these structures. So we use the technology that I mentioned in my prior talk, where we're going to do scanning EM and then shave sections at 4 nanometers a pop using a focused ion beam, and then do this through an entire cell. And this is what you've seen before, where we move through a whole cell using FibSim. We're now going to look at ER exit sites in this image. Now, in order for us to see the ER exit sites, we need a probe that identifies these ER exit sites, because they are not easy to see unless you know where they are. And so to do this, we used a cryocorrelative approach where introduced into this pipeline for producing the FibSim images is a transfer to a cryo structural illumination microscope. So basically cells that are on sapphire cover slips are high pressure frozen, and then that frozen cell is transferred to a scope of a structural illumination microscope that can gather images at cryo temperatures, because we do not want to take these cells out of a cryo state. We then image the cells using our fluorescence capability. The cells have previously been labeled with a probe. In this case, you can see here's the ER labeled, but we can introduce a probe that labels these ER exit sites. We take a picture, structural illumination microscope picture, and then we do free substitution resin embedding and then gather FibSim structures. Here is the cell that we're going to be looking at. This is the full FibSim sort of reconstruction through that cell. And we've superimposed our image, our structural illumination microscope image that was acquired prior to doing the FibSim through this cell, that allows us to visualize all of the ER exit sites that are labeled with Sec23 fluorescent protein. So these individual spots are all the ER exit sites within this cell, and we're now going to start looking at them because we can say, okay, this is the volume where the ER exit site is. Now let's look at what the structure is. So here are the individual slices through that volume.
Can I ask a quick question?
Yeah.
Just in your previous slide, it's maybe a little naive and very technical, but how do you actually do that alignment?
It's hard.
Yeah.
So that's very hard. That's a key thing.
Okay.
We put fiducials in this system. You can put little gold particles in just that you can see in both images. But it's challenging.
Okay. So
it's a critical step.
I don't know, maybe do you have any advice for people that are trying this not on maybe not as fancy microscopes, but just general alignment tricks? Is there something to pay attention to in particular?
Well, normally you would have you'd have fiducial markers that you'd introduce into the system for alignment. Here we're doing some other things too, like we do sort of it turns out that the cells slightly, they change their volume slightly when you do the free substitution. So we have, our computational people have done a type of warping approach that allows it to accommodate that slight change in volume that the cell experiences when it goes from a frozen state to a substituted state. So that you probably don't have to do if you're not, unless you're doing this particular approach. But yes, it's a very important, being able to align the SIM image with the FibSim image is extremely challenging. But we've been able to do that. And here is one particular, here's an ER exit site that we're now segmenting out. So each of the membranes are outlined in individual scanning EM images, and then they're summed up to look at the whole image in 3D. And that's what you're seeing here. And the interesting thing is, here's the ER, and here's this ER exit site, which is a tubule complex that associates with the ER at one or two necks. And it's closely opposed with microtubules. We've looked at hundreds of these ER exit sites, and their general diameter is about 360 nanometers, and the tubules that are comprising them are about 40 to 60 nanometers. And we know from our live cell experiments that there is communication between these two. This is a continuous structure emanating out of the ER, because we can frap cargo that has been loaded into this ER exit site and see recovery of the signal, meaning that cargo can actively move in and out of this structure as it is forming. It's being concentrated in some way. Okay, and this is just shown, sorry, yeah?
Can I quickly interrupt a quick question? Can you comment a bit on your choice of SIM as a light microscopy method? Is it just providing you with the proper sectioning and resolution to identify these sites? And I guess it's also a compromise between...
Yeah, I mean, you could say, well, why not do something like STED or PALM? So we could definitely do PALM. The key thing is being able to do this at cryo temperatures.
Yeah, yeah.
Okay, so STED we can't do at cryo temperature, but we can do PALM, or we can do structural illumination microscopy. And we chose structural illumination because it's easier, but you could definitely do PALM.
Because your choice of labels is also much broader, I guess, that plays into it. Okay.
Exactly. And I should emphasize that these fluorophores are actually brighter in a cryo state, which makes it easier to look at. So this is what the ER exit site looks like, sort of in a broad scale. Blue is ER. This is the ER exit site, and you can see these microtubules. One of the things we discovered, okay, somebody had asked in the prior lecture, what's surprising? This was surprising because previously from thin section TEM, people had proposed that what these structures were just clusters of small vesicles that had budded out from the ER by COP2 mediated vesicle formation. What we're showing is, no, these things they are not little vesicles that are clustered together. They're all interconnected into a single structure that itself is still attached to the ER. And furthermore, as shown in this slide here, these ER exit sites can actually swell, get enlarged, if you release cargo from the ER in a bolus. And that's what we've done to the right here, where we've expressed a protein, TNF alpha. Initially, it's trapped in the ER, so we can build up a large quantity of it. And then we can release it using what's called the rush assay by adding biotin. It now gets freed from its anchor that's been holding it in the ER. And now this molecule moves into the secretory pathway. This is 8 minutes after that biotin addition, and these ER exit sites swell. So instead of having more ER exit sites form when you have more cargo flushing out of the ER into the secretory pathway, the ER exit sites just get bigger. The container enlarges. And in our paper, we can describe precisely how big these guys get. We see an enlargement not only in the diameter, but also the tube diameters. So that then raises the question of what is COP2 doing at these contact site, at these ER exit sites, and also another coat protein called COP1, which has been it's a different type of coat protein than COP2 that's been speculated to be involved in recycling material back into the ER. And so when we used, in this case, ARIScan imaging to look simultaneously at COP2 and COP1 and a GPI rush cargo. So here's the ER. You can see this sort of tubule entity. And here's the ER exit site that has both COP2 as well as COP1 associated with it. What we're seeing is that the COP2 seems to be almost always more closely associated with the region of the ER exit site adjacent to the ER. And so we think that COP2 is potentially working at the level of this neck region, whereas COP1 is more peripherally associated. Many people in the field think that COP2 coated vesicles move through the cytoplasm to the Golgi, that they are the conveyors for ER to Golgi transport. So we looked at what happens to COP2 when cargo actually leaves these ER exit sites. So here's Sec23 that's labeling the ER exit sites. These are all the little ER exit site spots. And now we're going to follow what happens over time. So we're going to color code Sec23 at different time points over the course of about 15 minutes. You can see that COP2 does not move. It's staying at these ER exit sites. And that's in contrast to the collagen 10 cargo that we've introduced into this cell, where you can see it is actively translocating along these trajectories during that entire time period. So COP2 is not moving with the cargo as the cargo moves towards the Golgi apparatus. It stays behind. And that's in contrast to COP1, labeled with epsilon COP shown here. You can see if we map the trajectories of COP1 upon release of collagen 10 from the ER, you can see it's tracking with the collagen 10 containing transport intermediates towards the Golgi. They go from periphery into this Golgi region, and they are moving together. So let's look at these transports. And here's a cell that has been high pressure frozen 8 minutes after we've released TNF alpha from the ER. And this is a sim image of that cryofrozen cell. So we're now going to go in and start looking using cryocorrelative at each of these transport intermediates. Here's the FibSim cell. Each of these boxed areas are transport intermediates that contain the cargo TNF alpha. And what you can see is that these transport intermediates are purling tubules that have the cargo distributed across them. But importantly, they're not vesicles. they're not vesicles and they're not vesicle chains. They're a single tube that has the cargo distributed throughout it. And the tube is aligned with a microtubule. And in many cases, it's still connected to the ER exit site shown here in green. So here's the microtubule, here's the transport intermediate, and the ER exit site. And this is just, oops, sorry. This is a still image of that. Here's our reconstructed image where you can see the purling tubule emanating from the ER exit site. And this is the superimposed TNF alpha rush signal that was acquired using structural elimination microscopy. Now, this is not an artifact of overexpressing TNF alpha because if we just look at cells that have not been transfected with any fluorophore, we can see the same type of structures. Here are the ER exit sites, and many of these ER exit sites, you can see these long purling tubules emanating out of them that seem to be aligned or at least associating with microtubules, which we think are what are pulling these structures minus N directed towards the Golgi. So this, talking about surprises, this was like one of the biggest surprises for us. The idea that the transport intermediates are directly being pulled out of the ER, potentially on microtubule motors, and you get these purling shapes of these transport intermediates. COP2 remains at the neck of this whole system. COP1 is found at the ends of these tubes. It's found not only sort of at the ER exit site itself, but we can see it at the tip of these transport intermediates or in other sort of discrete domains. Why that is, we have no idea. But basically, this is our model for this ER exit site ultrastructural organization. If we zoom into the site where COP2 is localized, we felt it was really important for us to think hard about how COP2 might be working at this neck, because we know all the classic work of COP2 in vitro systems where it can assemble a bud through dynamic SAR GTP GDP exchange activity to draw the coat onto the bud and ultimately create a coated vesicle in vitro. What we're thinking is happening in these mammalian systems is that potentially because the membrane is being drawn into these regions so fast or dynamically that basically instead of a vesicle being created, you create this elaborate tubular meshwork. With COP2 continuing its cycle of binding and dissociation at the neck of this region and stabilizing that neck, actually acting as a gating mechanism for sorting into this ER exit site, which ultimately is going to interact with microtubule motors, potentially through ARF1 related activities that draws not only COP to this region here, COP1, but also other factors that potentially would allow this thing to start pulling along a microtubule towards the center of the cell. And with that, I just want to end by saying that merging these two technologies, light microscopy on the one hand with electron microscopy, there's tremendous potential for new biology, as I hope you can appreciate from what I've just said. Thanks.
Yeah, thanks very, very much. That was a lot of interesting things to think about, I have to say. So we are kind of closing up. Is there someone who would like to make a final question? Especially, I think we have some junior people that I would really like to give the floor to that didn't ask a question yet.
I'm not a junior person. I hope you don't mind. I'm not a junior person, but I have a question regarding ER during the cell cycle.
Yeah, ER during the cell cycle. Okay. Have we looked at it?
Oh, because that was my question. Because the nuclear membrane, as you said, it comes from the ER. So I was particularly interested in the way it's reformed at the end.
At the end, yeah. So
yes.
We and others have looked at that over the years, and just by light microscopy, doing live cell imaging. In fact, it was one of the first things that we did early on with GFP. Basically, at the beginning of mitosis, when the nuclear envelope disassembles, basically all that's happening is that it's being resorbed into the ER, which remains as a interconnected tubular meshwork throughout mitosis. ER does not fragment or vesiculate during mitosis. The nuclear envelope membranes are now, membrane components have now redistributed into this ER system. And now at the end of mitosis, what happens is that parts, the chromosomal material starts adhering to ER, and that creates the nuclear envelope. Basically, ER proteins that have affinity, lamin B receptor with lamins and other things, start diffusing and getting trapped into the areas where the ER has started adhering to this nuclear material. And with time, you can see the ER grow. And then as you start creating nuclear pores and pumping and trapping things, the system bulges out as an entire nucleus. There's a paper by Jan Ellenberg from my lab who did some live cell imaging of this in the 90s. If you're interested in seeing those movies, it was pretty exciting to watch this.
Yeah, I'll have a look. But are there any specific tethering proteins involved by any chance?
Of the nuclear material with
the chromosome, with the still very dense chromosome.
Yeah, all of those molecules that I showed you, there's like the sun tag proteins, there's lots of filamentous materials, lamins, that are in the nucleus that are interacting with the inner nuclear membrane to, it's a complicated interactive system that clearly is disassembled during mitosis, potentially through changes in phosphorylation status of many of those proteins.
Yeah, because just one more thing was, is there a signal coming from the still very condensed protonucleus telling the ER, come here, make an envelope around me? Or is it purely stochastic?
That's a great question. I think it looks stochastic. If you watch those movies, you just see slowly, we were looking at lamin B receptor, which gains affinity for the material that's associating with the nucleus at the end of mitosis. And you can just see it just slowly being drawn out of the surrounding ER into these membranes that are adhering to the chromosomal material. So why are you interested in that? Are you yourself looking at this?
Well, nowadays I'm doing computational biology, but I have done a lot of microscopy, and I was looking at cell cycle, and with particular interest for RNA polymerase binding to condensed chromosomes, which they do. I saw quite a lot of binding, even on the metaphase plate. But as I was progressing along the cell cycle, I just wondered how the nuclear membrane is starting to form. So at late anaphase, I have a lot of very condensed protonuclei with enormous RNA polymerase 2 binding. So at that stage, I was not only looking at the polymerase, I started looking at also the nuclear envelope. So I was
Yeah.
just wondering how that is actually formed.
Yeah. It's just pulled out of the ER, basically.
Yeah.
With some adhering molecules.
Okay, thank you.
Yes, thank you very much. Maybe this is very interesting. Maybe I just want to again ask one more last junior person to ask a question, and if everyone is sort of completely hypnotized by everything we've seen, then can of course understand that too. So maybe I just want to take the opportunity to thank Jennifer again. I think it was really, yeah.
Could maybe just ask one question?
Yeah.
That's okay. Yeah, it's just totally mind-blowing stuff. Thank you. It's amazing talks. But I was blown away by these pearl tubules that you find that go towards the center of the cell. I wondered how common that kind of morphology is in cell biology in general, that you see these long tubes with bulbs in the middle like that. And I guess from your description, I understood that they're being pulled along. So does that mean that tube is getting longer and longer, or does it just break off of those exit structures and just kind of go along sort of treadmill?
So these are great questions that we don't have answers to. We think that because of the periodicity of the bulls, this could be something related to purling instability that's been described in the biophysical community for, when you pull tubes. But the question you asked in terms of how general this is for other membranes, I know that there's been some reports of endosomal membranes showing this type of purling structure. But importantly, I just want to emphasize how hard it is we really haven't had the technology to see these structures until this kind of whole cell reconstruction. Transmission EM is frequently just a small sector of the cell that people are looking at. And they're not looking at 4 nanometer isotropic resolution. They have great XY resolution, but they're not doing isotropic 4 nanometer. And certainly the tomography can definitely get there, but the tomography is small subsectors of the cell.
I'm also wondering if those structures might be less stable in conventional fixation techniques. And
Yeah, absolutely. That's another issue.
Yeah.
I saw somebody who had a hand, Joachim Schmidt. No, okay, whatever. Okay.
Joachim was actually waving.
Sorry, I was waving at Yuki's kids.
Okay, cool. Good.
Sorry, my cats were running in.
Right.
Great.
Yeah, I don't want to. I want to give people more option to ask questions, but I also have somehow some kids that I have to feed. So I would maybe I can just again thank Maaike very much for her nice talk, and also Jennifer. It was very, very stimulating. I very much enjoyed it. I hope the rest did. So what thank you all for coming. We will let you know again when we will have our next seminar series.
Great. Well, thank you for inviting me and participating.
Thank you. It's amazing.
Thanks a lot.
Thank you very much.
Bye.
Bye.